Thank you very much, Stephen, and for this very kind introduction. And thanks to Stephen, Christian, Felix, and the whole EVOLTREE team for inviting me. So let's start sharing my screen. Great. Yeah, so Stephen has already introduced me, so I don't have to repeat that. It's a great pleasure today to talk to you about how we in my group try to use real-time genomic approaches to better understand One Health. And I guess specifically within this EVOLTREE seminar that's focusing on new technologies for evolution and genetics, we specifically, I thought I would focus on how this real-time genomic technology can really open new avenues for us to understand like different health dependencies better. Now, let's start by actually defining what One Health means, because I do from time to time experience that a lot of people haven't heard of this concept that you might have also heard of in terms of planetary health or eco-health. Yeah, so there are very different names for it, but the general concept always describes that all the health on our planet is inextricably linked. So it extends the health concept that often for us focuses on human or global health to the health of other organisms, and that can really range from microorganisms to other vertebrate and also plant species, for example, and also to the health of entire ecosystems. And One Health research really tries to understand the interdependency and the interaction between these different health system interfaces. So by seeing this, I'm sure that like many of you could define your own research as One Health research as well, right? I think the whole concept just tries to really put an emphasis more onto that there are connections between these different health systems. And I think maybe especially for the EVOLTREE community, what is a really good example of how these health systems are interconnected is how anthropogenic impacts on the environment lead to habitat destruction. This has consequences for biodiversity loss on the ecosystem level, on the species level, on the genetic level. And that again can actually lead to increased zoonotic risk and also other human health risks, such as antimicrobial resistance. Now, this is like a sort of interdependency that has actually been well described even by theoretical scientific literature already. For example, through the concepts of mixing vessels and the dilution effects. And I can really recommend those two papers to you if you are more interested in the background. But what that basically describes is that if we have a healthy, biodiverse environment, we don't allow pathogens to evolve so easily and also to spread so easily. So the mixing vessel effect specifically says that if we actually provide like very uniform populations, so a very dominant species, for example, that in addition have very low genetic diversity, such as it often occurs in livestock, for example, we are providing like basically a breeding ground for pathogens that can mutate quickly, that can share genomic material, and that can really, acquire all sorts of genetic variation to then spread to other species, to be particularly virulent, and so on. The dilution effect is like a very similar concept, I would say, but it more relates to the transmission of pathogens. Yeah, that if you think about a highly biodiverse ecosystem, we are actually basically introducing suboptimal hosts for a pathogen that don't allow it so easily to spread than if we had like a very biodiverse, poor ecosystem. Yeah, and this is sort of a bit the framework that I would like to place my talk in today. Obviously, I guess the past or ongoing COVID-19 pandemic has been a really good example for these connections between the health systems and the connection with biodiversity loss. And we do know that most likely the SARS-CoV-2 pathogen has actually originated from like close contact with wildlife and a zoonotic jump over. So now, to give you a brief overview of the work of my group, so what we are trying to do is to use genomic approaches and artificial intelligence or AI or machine learning approaches to better understand One Health. And as you can see here by the lab members, we are really working across all these different health systems using similar technology in the space of genomics and AI. So it really extends from really biodiversity research to ecosystem health monitoring to more like human health and zoonotic risk research that we have over here. And today, I would really specifically just focus on the real-time genomic research that is happening in our group, because I think it provides a really nice framework to actually understand these interdependencies between the health systems that I just mentioned. So to define the second word in my title, real-time genomics, when I talk about real-time genomics, I'm really talking about nanopore sequencing technology, because it's the only way to do real-time genomics at the moment. How nanopore sequencing technology works is that we have tiny nanopores embedded in a membrane, and we have an ionic current going through these nanopores. Now, if either our single-stranded DNA, as you can see here, or our RNA, any sort of nucleotide, passes through these pores, it's disrupting the ionic current in a characteristic manner. And this results in the so-called, sorry, I have to show that first, in the so-called squiggle data, or like ionic current disruption. Yeah, so like a really dirty signal of this ionic current that we can then, however, translate into the actual DNA sequence using bespoke base calling algorithms that are also mostly based on AI, on deep models. Yeah, because assuming that a specific base combination in these pores is going to disrupt the ionic current in a very specific manner. And this is a really exciting technology that gives us a lot of advantages over other genomic approaches. And I'm very happy to say that we are detailing especially the advantages of this genomics research in the context of One Health in a recent review article, where we are also really trying to create a perspective of how real-time genomics can be used in the context for One Health in the future. So what are these advantages now that we try to leverage? Firstly, so what we can do with this approach, we can really sequence the native DNA that we just extracted from our sample. And this means that we can get very long sequencing reads, basically as long as the original DNA fragment was. And when we think about actually trying to get any information from this genomic data, like species origin, or even de novo assigning of a species, then these long reads can really help us and make our results more robust. Further, we can create data, as the name already says, in real time, because at the very moment that this ionic current is being disrupted, we can actually base call it into the original DNA sequence using powerful AI models that we can implement on really powerful and fast GPUs. Yeah, this means that actually our time to result is getting really, really short. In addition, we can use this real-time sequencing for so-called selective sequencing, where we, at the very moment that our DNA fragment is still in our nanopores, we can start base calling it, map it to some reference data of interest, and then based on if the first data that we are receiving is mapping or not, decide if we want to continue sequencing a specific DNA fragment or not. And like this, actively enriching or depleting for signal. The third advantage is that we can use this, what I started calling the power of the squiggle, for other, for inferring other sorts of characteristics of our genomic material than just the DNA sequence. I think the best example here are epigenetic modifications that, as you can see here, will obviously disrupt the ionic current in a very specific manner as well. And we can leverage this information to then actually get all sorts of epigenetic modifications from the raw data that we are retrieving anyways. And this is like the calling of epigenetic modifications is really something that the nanopore community is using very regularly nowadays, but there are other examples of how we can use this, and I will very quickly present some of this research at the end to at least give you a bit of a hint into our AI research in the group. And finally, the entire approach genomic approach is portable. Yeah, so especially if we sequence, if we combine the portable sequencing device, for example, the Minayan here, with a portable, powerful compute, like these embedded AI machines from NVIDIA that we have been benchmarking, so that you can actually do the data creation and analysis directly at the point of interest, if it's now in the field for conservation questions, or, for example, directly in the clinical setting. Now, right, taking a step back, what I would like to show you today are like a few examples of how we can use real-time genomic approaches to better understand One Health. And I would really hope that it might maybe also develop some ideas for your own research into how you can either venture more into the One Health space or actually use these real-time genomic approaches. But I think what is this really powerful way of like using genomics here is that we can actually obviously create the very same data from like a lot of different sample types that we can get from all these health systems that I've just introduced to you, and like bring them into connection, yeah, by analyzing them in the very same manner. So let me show you a few examples here. So firstly, like throughout my PhD and my postdoc, I've started using nanopore sequencing technology for metagenomics. Now, really briefly, because I didn't have my my own career timeline today in the talk, because I don't think that's so interesting for you, but like I'm really coming more from the computational statistical genomic field, but have at the same time done like a lot of ecological field work in my career. So I sort of very much enjoy the both, and the laboratory work a little bit, as long as it's not too often. But I want to say this because I guess you have to like look into this sort of research from both like thinking about establishing it in a practical manner for any sorts of monitoring purposes, but also looking into the computational challenges that we might have. Yeah, so during my postdoc in New Zealand, I actually helped establish real-time genomics in low-income countries during the COVID-19 pandemic, making really use again of this portability of the approach. And I've also helped establish microbiome sequencing in a small gynecological clinic. Yeah, like again, like making use that we don't need like big sequencing machines to allow for real-time genomic approaches. I've also like helped establish the wastewater surveillance framework in New Zealand during the time, and also worked on actual in situ biodiversity monitoring, yeah, using like very similar approaches. And I will briefly talk about this research in a bit, as well as about actually surveying pathogens in endangered wildlife. How I very originally started using nanopore sequencing technology was, however, for like actual sequencing ecosystem monitoring, for sequencing freshwater samples. Yeah, where we were interested in actually like very early on when the nanopore sequencing technology came out, to explore how robustly it could assess freshwater quality. And that was a side project during my PhD, which was actually like very much focused on statistical cancer genomics and single cell genomics and stuff like that. But I got very interested in this technology. So we teamed up with a group of PhD students, and that was back during my time at the University of Cambridge, where we just decided to use our local river, the Cam, to establish like a freshwater monitoring protocol. And what we did is we took like several water samples at different times, at different locations across the city of Cambridge, where upstream of the city we have a quiet natural habitat, and then we have like a quiet, a polluted area that is heavily being used by humans in the middle of the city, and then a bit more of a natural habitat again downstream. Also notably, we just sampled just downstream of a sewage pipe outlet of a close by wastewater treatment plant to see if we could find any specific signature in the water there. And I will just show you like one result of this study that like sort of, I think, really nicely explains how this real-time genomic approach can elucidate the One Health concept. Now, what you see here on this figure is you find all sorts of potential microbial pathogens that we found in the freshwater samples here on the y-axis. And here you actually see potential or bacteria that we know are being used for the treatment of wastewater, yeah, so in the close by wastewater treatment plant. Now you have here on the x-axis our gradient of the river, like along the city here, with like the upstream natural environment, the more polluted urban area, and again like a more natural environment downstream. And here, I think the most important part to look at is like actually the red coloration here, which represents the relative enrichment of any of those taxonomic groups across these different sampling sites along our river. And I hope you can appreciate that we found like a relative enrichment of potential pathogens at location 7 here. And you have to know that location 7 is really like the most polluted location in Cambridge, where you have like a lot of impact by human usage, by houseboats, by agricultural intakes, a lot of cow meadows around and so on, which was something really interesting to see, like, and basically made us hypothesize that anthropogenic impacts on the environment can really have potential human health repercussions, right? By like allowing potential pathogens to flourish. Now at location 8, that was the sewage pipe outlet that I already pointed you to, we indeed were able to find like this, like really nice signature of wastewater associated bacteria, which I guess made sense, right? Because they had just been used in the wastewater treatment and were then being let into the river through this pipe. But it like really for us showed that this approach seems to be working very well and like capture like major ecological changes in along this river gradient. What we, however, also found was an enrichment of Leptospira, a potential zoonotic bacteria that can lead to the zoonotic disease leptospirosis that can even be deadly for humans when left untreated too long. And that was like really interesting because in Cambridge, we do know that people present with leptospirosis in the hospital from time to time, often, as doctors told us, also undiagnosed. And obviously, like the fresh water that is being used a lot for rowing and swimming and so on could be a very reliable source for it. But it was really interesting for us that this Leptospira signal was so enriched just downstream of the wastewater treatment plant, making us hypothesize again that actually the anthropogenic impact through this wastewater treatment outflow had an impact and allowed this potential zoonotic bacterium to flourish, with obviously potential human health consequences, again, like really nicely illustrating the One Health concept. So with this, I thought I would briefly talk a bit about my biodiversity-focused research during my postdoc in New Zealand until beginning of last year, because I also thought that the EVOLTREE community in general might be quite interested in that. So I said I moved to New Zealand to try to really use genomics approaches to better understand how we can use this for conservation of critically endangered species. And I worked on a variety of different species in New Zealand. My main project was actually on the takahe species, a critically endangered rail species that is also flightless, and that is just actually increased to a population size of more than 500 individuals again, but still like really endangered. And where I was basically establishing like an entire protocol for a conservation genomic project, from funding acquisition to actually field work and taking the blood samples to laboratory work, computation analysis, and also partially the development of certain computational tools that we needed. Now, I think the takahe is a really nice example of the actual the wildlife in New Zealand in how it has actually become so endangered, because for a really long time New Zealand didn't have any mammalian predators until first human settlers, both Maori and European settlers, actually introduced them and also started in addition destroying the habitat, so that actually, you know, like especially flightless bird species like the takahe were like suffering a lot and were decimated like very extremely in numbers. So this paper we are actually currently wrapping up, and I'm hoping to provide that to you very soon at the beginning of the next year. But today I thought I would talk a little bit about my research on the kakapo, another critically endangered flightless bird species. In this case a parrot species that is actually down to only 248 individuals nowadays, and has even in the 1990s been reduced to only around 50 individuals. Which means, right, if you already hear this sort of story, and I'm sure you all know this very well, we have a lot of problems with this species in terms of inbreeding and low genetic diversity. And this low genetic diversity obviously extends to all sorts of immune genes. Now we can use genomic approaches. If we go back to our thinking about how we can use genomics for conservation in the context of One Health, we can use this to actually understand the host or the actual fitness of the critically endangered species itself. Yeah, and I'm just quickly going to point you to some published papers here, but then we'll talk about some more unpublished research. So firstly, we created genomic data of nearly the entire kakapo species, and then basically really stringently assessed the inbreeding and genetic diversity that is left in this species. But we also tried to actually assess the genomic diversity of the kakapo in a non-invasive manner, just using environmental samples. And we've also done a lot of quantitative genomic studies where you can actually associate the genetic diversity that is left with any sort of phenotypes that might have fitness consequences, and also with phenotypes that actually relate to the microbial load or microbial diversity itself of these species, which might also very much be like a fitness output. Now, I said I wouldn't talk too much about this research today, but please ask any questions about it afterwards if you like. What is in addition obviously the case for critically endangered species, in addition to their inbreeding and low genetic diversity, is that we have very few individuals left, and that we often have highly connected populations. In the case of the kakapo, it's only a few populations that live on tiny offshore predator-free islands in New Zealand. And all this together means that there is a really high risk of final extinction through any sort of catastrophic event, such as, for example, infectious disease. Yeah, and I guess here you already see, again, this One Health concept coming into how actually the health of the species is obviously also defined by the health of its environment and the health of the presence and evolution of certain pathogens around it. In the case of the kakapo, we actually have experienced regular aspergillosis outbreaks in the newly hatched chicks. The problem with these aspergillosis outbreaks that are caused by a fungus Aspergillus, it's also quite common human disease, is that the infection was of unknown source and of an unknown really epidemiological network, so how the disease would actually spread. However, the conservationists suggested that there was a connection with warming climate, which means that the disease is probably there to stay. Yeah, so we really needed to have a quick in situ monitoring approach to profile the Aspergillus that we could find in the chick oral cavities that could potentially lead to an infection, and to specifically detect the highly pathogenic Aspergillus strains that we expected to create these infections. So what we established directly in situ in one of these remote islands in New Zealand was a pathogen monitoring approach, where we would go, because the kakapo is a nocturnal species, at night to the nests of the kakapo when the mom was actually out foraging, swap all the kakapo chicks, and directly in the field extract the DNA and start the nanopore sequencing and analysis. And like this, we could actually find evidence of Aspergillus in the saliva. In our entire sampling efforts in 2022, we only found evidence of non-virulent Aspergillus in the saliva, but also none of the chicks subsequently developed aspergillosis. So we are actually going to repeat this in subsequent breeding seasons. But the very important part about this approach is, and that's why I wanted to showcase it to you, is that we can make this approach fully portable. We paired the sequencing device that you can see here with a portable battery, this embedded AI machine that does all the heavy lifting analysis for us, and a portable solar panel, so that we can really directly in the field create and analyze our data. And the most important part about this project was actually to train the conservationists in this approach, so that they now in the next breeding season can try to use it themselves and find any evidence of highly virulent Aspergillus. Again, I think really think about a nice approach is how we can use one technology that we've established on the island now to monitor a variety of different things. If it's now the genomic health of the species itself, such as inbreeding and genetic diversity, as well as any sort of pathogen that could show up. And it's so important to be able to do something like this in situ, because until you would have brought any of these samples of the islands, you would, basically not be able to adapt your management to this anymore. Yeah, so the time to result would just be far too long. Great. So with this, I thought I would actually show you some very recent results from my own group now that we are also currently wrapping up as papers. I think with that, then I will have given you hopefully a bit of an overview of really like the very different extremes of how we can use real-time genomics for One Health, and then we can discuss more afterwards. Yeah, so the first approach is actually the first project is about real-time air surveillance using real-time genomics. That's a project by PhD student Tim in collaboration with the IS Global Institute, Global Health Institute in Barcelona, where we were thinking of, when you think back of our water monitoring project, extending this sort of ecosystem surveillance monitoring to the actual air, because there's actually very little that we know about the air microbiome or bioaerosols and how they are impacting human health in the long term. So I'm very happy to say, and I have to go a little bit quicker here right now, but very happy to say that we have actually managed to establish a protocol that just uses nanopore shotgun sequencing for air microbiome assessments. So with shotgun sequencing, I really mean we are just sequencing the native DNA out of these air samples without any sorts of amplification or targeting of marker genes. And here I show you the results of like one of our then first experiments in a controlled environment, which was basically the greenhouse on our campus, where you on the y-axis see the relative abundances of different microbial taxa, the most abundant ones visualized here. And you see these air microbiome measurements across consecutive days, really showing you that we can very reliably and stably assess the air microbiome in this greenhouse. Now we next ventured out into the actual into an actual natural habitat where we obviously expected potentially larger fluctuations across days between the air microbiome. But yeah, we were very positively surprised that we could also detect a quiet, stable air microbiome across consecutive days in this natural habitat, which was just like a more natural area close to our campus. And that even in the urban area, we have very small sample size here until now, and we are creating more data right now, that we can also relatively stably assess the urban air microbiome. And most importantly, we can show that we can distinguish really ecosystem-specific air microbiomes between these locations. And as you can see, they all really look like relatively or quite different from each other, so that we can then really think about human health consequences, or at least functional consequences of these air microbiomes. The exciting part is also that despite our data, it's like air microbiome data, it's highly fragmented DNA, but we were still able to build quite ugly de novo assemblies, I have to admit, like if some of you don't know, but like a very nice de novo assembly would just be like a nice bacterial circular genome, which we definitely don't achieve here, but we can achieve a de novo assembly per se, which can help us like also redefine new microbial species in our environments, especially when it's an environment like air that has actually not been probed a lot in the past. And we can then use this assembly to functionally annotate it, as I just said. In this case, for example, for the Sphingomyces bacterium, we found evidence of functions for styrene side chain oxidation, which amongst others can help microorganisms to digest microplastics, which was really interesting because we, in this really preliminary data, find like a clear enrichment of Sphingomyces in more polluted areas in the urban area, letting us hypothesize that Sphingomyces might be a biomarker of pollution because of its potential ability to degrade the polluted matters such as microplastics. And we can also do all sorts of other functional annotations, right? For example, in Streptomyces, we found an antimicrobial resistance, vancomycin resistance, which can also be like really nice to think about the actual functional consequences of the air microbiome. And as I said, we are currently extending this research. So PhD Tim and postdoc Albert, who you see here, have just been in Spain at different sites for some monitoring efforts to both further assess the air microbiome in urban areas, specifically in Barcelona, across different anthropogenic gradients, as well as for directly targeted pathogen monitoring in the wild, where specifically Albert, and I can't talk about this project today, but he's like a proper avian influenza virus expert, and he has like established all protocols now to detect avian influenza in these air samples, and has actually been really successful in his first fieldwork. So at whatever talk I will give next, I'm sure I will be able to talk about these results as well. Great, yeah, so that was like another example of like ecosystem monitoring with assessing potential human health consequences. Now I would like to jump to directly human health consequences, because we can also use real-time genomics really directly in the clinic. And this is research led by PhD student and medical doctor Ella in collaboration with like a lot of international collaborators. So here again, we are making use of our long sequencing reads to actually predict antimicrobial or antibiotic resistances, because we have like these big gene clusters that often involve several genes, and our long reads actually allow us to potentially have all these genes on a single read, really robustly allowing us to predict certain antibiotic resistances. Further, I've already talked about the portability of the project, so we could really think about bringing this real-time genomic technology directly to clinics for them, to help them with antimicrobial resistance profiling, and especially the low upfront investment into this technology. Yeah, because the sequencing machines per se basically don't cost anything. Yeah, you only have like the running costs of the consumables. Will potentially allow us to increase global accessibility to any sort of antimicrobial resistance profiling in comparison to what we do now. Because when you look into like current approaches, and this is like just as an example for bloodstream infections, like the current phenotypic approaches that are being used in the clinic are, you know, like based on the species identification on so-called multitoph, which is basically mass spectrometry, and the resistance prediction on Vitek-2 approach, where you really just experimentally subject all your bacteria to different antibiotics. And for those alone, the machines are really, really expensive to establish, yeah, like really all around the world. For example, if you think about low and middle income countries. And here, like a cheaper approach that actually relies more on like running costs could be very, very helpful. And I guess importantly, like coming back to the speed again, we definitely, and Ella has illustrated this really nicely here, can see that phenotypic, that the real-time genomic approaches can definitely be faster than the phenotypic approaches, which can really take up to several days in certain cases. But the question obviously remains if a genomics-based approach can be as accurate as a phenotypic approach. Yeah, because obviously genomics approaches will always be predictions. They can't really replace an actual phenotypic test where you are subjecting a bacterium to the antibiotic. And also to quickly put this research into a broader context, like understanding this will obviously also allow us to analyze all other sort of samples, all other sort of data that we are creating right now in a broader context, thereby understanding how accurate are these functional predictions that we are getting from such genomic data. So Ella specifically is creating like a huge catalog of genomic data of different bloodstream infections right now that are matched with the actual phenotypic predictions, like really providing us like an exciting opportunity to benchmark like how well real-time genomics works at the moment. And I'm very excited to now present one specific patient case to you. So this patient presented to Ella with a bloodstream infection and with fever, and a blood sample was taken, and the patient or the infection actually with this, again, right, experimental approach showed to be resistant to carbapenems, antibiotics, because as a follow-up approach showed, of a so-called KPC resistance gene, yeah, that is like conferring resistance to carbapenems, but was predicted to be susceptible to the last resort antibiotic ceftazidime avibactam. So because of this last resort antibiotic was administered to the patient. However, the patient's status deteriorated, and when another blood sample was taken, the actual infection had completely turned around its antibiotic resistance profile. It was now susceptible to carbapenems and resistant to the last resort antibiotic, which is actually a quite severe resistance to find in a clinical setting. Now, while obviously antibiotic treatment administration was adapted and changed, the patient had unfortunately already deteriorated too much and passed away a short time after. But this was like one of the cases that we had access to create our genomic data and dig into a bit deeper. And I'm showing this to you because I think it's a very beautiful case of what we could potentially achieve with real-time genomics in the clinic. So let me show you our data. So firstly, for the second blood sample, our genomic data can replicate the clinical results, and that we are finding susceptibility to carbapenems because we don't find any evidence of any resistance genes, and resistance to the ceftazidime avibactam because of a so-called KPC14 gene. Yeah, I've just told you that KPC resistance genes are conferring resistance to carbapenems, but KPC14 was actually one of the novel ones, the few KPC genes that has been found that actually confers resistance to ceftazidime avibactam and susceptibility to carbapenems. Yeah. So here are both our approaches basically agreed in the prediction. Now, for blood sample 1, we could also confirm the resistance to carbapenems that was found originally in an experimental setting because of these typical KPC genes, yeah, that are conferring resistance to carbapenems. Now, what was interesting, that is one KPC14 gene that here allowed us to predict the resistance to ceftazidime avibactam was already there in this sample with just a single copy. And while we would have obviously not trusted like a single copy in our entire data set to predict any sort of resistance to ceftazidime avibactam here, we could have really made use of the real-time genomic approach in the clinic that would allow clinicians to basically just run your sequencing a bit longer and just create a little bit more data, analyze it in real time, and see what you can learn from your increased data amount. Now, for this specific case, we simulated and ran the sequencing for another 5 hours, and we could indeed find 4 more, so altogether 5 copies now of the KPC14 gene, which would have meant if we had real-time genomics established in the clinic at this point, we would have learned about the ceftazidime avibactam resistance a lot earlier than in the actual clinical case, and that could have potentially saved a patient's life. We've also done a lot of follow-up computational studies, and we are currently publishing this as a brief communication, actually, so you can dig into this a bit deeper if you like. But what we've basically found was that this KPC14 resistance gene was present on a low abundance plasmid already in this blood sample. And probably then the administration of the ceftazidime avibactam had such a strong selective pressure, right, onto this microbial community that this plasmid had a huge selective advantage and actually became more dominant, and also phenotypically led to the resistance in ceftazidime avibactam. So I hope that this really shows you a bit how real-time genomics can not only be used, right, in like the more conservation and ecosystem setting, but also in the human health setting. So now, briefly in the end, I would like to, as promised, talk to you a bit again about the power of the squiggle. And yes, I still have several minutes, so that's great. Right, so maybe quick breathe through, because I know I'm rushing over quite a few projects here, but I hope that this would give you a bit of an overview of the sort of research that's possible with real-time genomics for One Health. So, but like thinking again back to the power of the squiggle, right? If I can remind you that the squiggle is the actual disruption of the ionic current while the nucleotides are passing through the nanopores, and they are decoding information about the DNA sequence, but also information about potential epigenetic modifications. Now, in a lot of the approaches that I've shown you, when we do this shotgun sequencing or metagenomic sequencing, we are just sequencing the entire DNA of microorganisms. We often have the problem, because we are not actually culturing these microorganisms, that we don't know if the microorganisms that we detect from DNA were actually living alive and virulent at the point of sampling, which is a big problem of like metagenomics in general. So if we think about the functional consequences of microbiomes, we would ideally only like to assess the microorganisms that are actually alive. Now, I had the a little bit crazy idea that we could potentially like leverage this squiggle signal that I've been talking about to infer if a microorganism that we have sequenced was actually alive or dead at the point of sampling. Assuming that as soon as a microorganism dies and its DNA gets exposed to the environment, to increased temperature, UV, humidity, and so on, it acquires a maybe specific signature that we can actually find on this squiggle data. So I asked our PhD student, Harika, who is actually doing all the AI heavy lifting in our lab normally, to like grow bacterial cultures, like just as a first easy experiment, and kill those cultures or let them live, and then nanopore sequence them to create the squiggle data. And we have then like benchmarked a lot of different AI models or deep models onto this squiggle data to predict if the DNA came from a dead or from a living organism. And just a few weeks ago, we have had a breakthrough where we actually are now able to train a model, specifically a residual neural network, for those of you who are interested in that can, with quite good accuracy of 82%, predict if the DNA came from a dead or from a living organism. And now I obviously have to say that this is a first experimental study with like very extreme data. So to now transfer this to like a more natural environment, we need to do a lot of follow-up studies. But the cool thing as well about is actually using AI models for that is that we can use subsequent so-called explainable AI methods to better understand why our model is actually making these predictions. In this case, we use so-called class activation mapping, and I won't go into detail about that. The important part is that we can visualize, and that you can see here by the red areas in our squiggle data, what sort of signal change is allowing the model to make its prediction correctly that the DNA came from dead or living organisms. Yeah, and we are currently interpreting this with a lot of other metadata. We might have some hint that it indeed has to do with epigenetic modifications, but we are currently following up on this, and then also really would like to publish this very soon to also make the models available to the community. But yeah, it's looking very, very promising. And I said, like, that's obviously just the first step, but very happy to discuss what the next steps would have to be for such a project to also work for the community for metagenomic assessments. But I really like to present this result at the very end of my talks at the moment, because I'm also thinking of that this could this power of the squiggle, right, could really open a new avenue of different sorts of research in the genomics field. Because, for example, just to present you some ideas, what we are using here right now, right, basically this signature of degradation to predict the viability of microorganisms, we could also use in other settings. For example, maybe to actually predict the gene usage, so how often a gene is actually being transcribed in your genomic data, or certainly to predict if the DNA, your ancient DNA was actually of an ancient source, or maybe was like contaminated by a modern source. We can also use this squiggle data very differently to infer the taxonomic origin of our DNA, and that has been done by others as well already, so we haven't invented this. But we have recently trained a transformer. This is like one of these AI models that are normally being used for these large language models like ChatGPT that you hear about everywhere right now, to really, really get a good accuracy of 96% to, in this case, distinguish if DNA came from human or bacterial DNA. And this is really cool because it gives us an orthogonal source of information of the taxonomic origin of DNA, which, as you will know when you are in this analysis yourself, often is not actually so clear, right? We might have mismappings to databases. We might have quiet, dirty databases, right, that are actually, for example, incorporating human DNA into bacterial assemblies and so on. And so actually having some more proof that what we are analyzing right now is, for example, really human or from a vertebrate from a fungus or from a bacterium can be really, really helpful information for our analysis. And I've had some other ideas of how we can use this squiggle data, but these are really just hypotheses at this point. So I have no evidence yet that this would work. Potentially these factors that I'm showing, will also leave a specific signature on the squiggle signal, such as, for example, and this is something we are actually currently doing, PhD student Samir in our group, to predict the isotope load of DNA. But also like completely other things, like, for example, predicting if a microorganism was present as a biofilm, if a phage that we think we sequenced was actually a phage or a prophage, or for example, if a resistance gene was located on a plasmid or on chromosomal DNA. Yeah, so we can, like, for all of those things, test if we actually find differences in squiggle data, train models if we have clean training data, and actually see if we can leverage this information in addition for making, for really saying something about the functional consequences of the microbial communities we are measuring. So I hope with this I've shown you a little bit of the research directions of my group in Munich. I would really like to acknowledge all of them. I've been really lucky with them. It's a fantastic group, and really please be in touch if you're interested in any of these projects in particular. And then I'm very happy to put you in touch. And very happy to discuss with you now, any of the specific projects or how it might even have some relevance for your own research. So thank you very much.
That's great. Thank you very much, Lara. Fascinating stuff, and really, I'm sure is going to stimulate a lot of interest. I'll just say thank you very much for the talk. That was superb and really what we were looking for, and ideal for this series where we're just really looking to get a flavor of what people are doing in other fields. So I will open it up now and just ask. There were a few hands up immediately, but if anybody has any questions or would like to kick off the questions, then please let me know now.
Thank you very much for your nice words, Stephen.
Okay, Felix, thank you. Please go ahead.
Someone has to break the ice. I see this snowy landscape here. Thanks, Lara. It was really intriguing. I have a very naive question regarding your last topic. Could you potentially also use or see for the presence of RNA in your samples to see if there's gene expression, and that would be an indication whether the potentially pathogen organism was still active?
Yeah, very, very good question, Felix. Thanks. Yeah, using meta-transcriptomics, so basically just sequencing the RNA of any sample instead of the DNA, can indeed give you a very good hint of what has just lived in your sample, because RNA, and Felix will know all of this, but just for the community, RNA can be degraded. It is degraded a lot more quickly than DNA, which makes it actually more difficult to analyze and to extract and everything, but which can be leveraged as an advantage when we're thinking about, it's degrading very quickly in the environment, which means if we find evidence of it, we do know that the microorganism was just present. So firstly, I think, but that's more of a practical thing, is I think that actually RNA extraction and sequencing is still a little bit more difficult, right? For RNA extraction, because it's degrading so quickly, you have to be really careful during the extraction. You have to do it on ice. For actually sequencing, you could do direct RNA sequencing, but it really doesn't work as well as direct DNA sequencing yet with nanopore technology. And besides that, you could do conversion into cDNA and sequence then, but for that you would obviously introduce certain biases just during conversion and amplification, and you would obviously need a PCR machine if you wanted to do it in the field, let's assume, and so on, right? So but that's just on the practical front. Now on the more let's say theoretical front, what my problem is with meta-transcriptomics is that obviously the RNA degrades so quickly. Yeah, so that you might actually, really just get a snapshot of the current functions of an ecosystem. Whereas, I think the question is a little bit like, what time frame are we sort of interested in, right? Like, because if really just something has died, maybe we might still be interested in it, right? Because it still lived and flourished in this ecosystem, but very often it's not easy to still find something like that in the RNA. Whereas with the DNA, and that would be the goal of this study, is to develop a quantitative predictor of basically degradation, and that we could say, hey, this, instead of this binary prediction, what we are doing right now, to predict quantitatively what's the level of degradation, how long has this DNA been exposed. Like taking the example of the freshwater study that I showed, right? We would assume that maybe for the wastewater bacteria that have been already killed off in the wastewater treatment plant, the degradation would be like really, really high. And we could see, these have been killed off with like really extreme methods. Whereas for something that has actually just died in the environment, we would see a much less signal. Yeah, so that's sort of more of the vision and that I would have. Whereas with RNA, I would feel we would like just miss a lot of the taxa that were actually there because it would degrade too quickly. Plus, you would never get the entire genome, right? You would always just get certain genes.
Yeah. Okay. Thank you.
Yeah, thanks. So you realize I could talk about this a lot.
A comprehensive answer. Thanks, Lara. Anyone else? Another question? Christian, yes.
Yeah, one of the hosts, I should not ask too many questions. I have quite many, but my first one is actually, you did not really talk a lot about the DNA extraction that you do in the field. Can you say something about that? Is it that easy as you say, or is it easy for certain organisms?
Yeah, so thanks for bringing that up. I didn't really have time to talk about this today. We have also worked on automating all of these processes more. So actually also like the library preparation and everything, but like also specifically the DNA sequencing. So in New Zealand, I've used one approach for DNA extraction automation. And now in the lab in Munich, we are trying to use the biomeme approach that many of you might have heard already about, which is like a sort of in-field DNA extraction approach. So there are ways of trying to simplify the DNA extraction to directly do it in the field. Now, with that, often the problem is that you still have a lot of contamination in your DNA. And if you actually want to sequence your native DNA on your nanopores, you will run into issues because the contaminants are going to block your pores. And that's why, like practically in many of the projects I've shown you, we are still using highly optimized QIAGEN DNA extraction kits that we are just employing on a portable bento lab, right? I mean, in the end, you only need a centrifuge and yeah, actually, I mean, yeah, for the QIAGEN kits, you really just need like a centrifuge and so you could just take a portable centrifuge with you actually to do these approaches, which obviously means you still have to do a bit of pipetting in the field. There's risk of contamination. So it's not what we want to do. So we are actively always benchmarking other approaches, but it's always a lot of work for any sort of new sample type you put in. So but I think once you have an established approach, there are a lot easier ways of getting the DNA out as well. But so in all of the in-field approaches I've shown you, it was like bento lab plus QIAGEN kits.
Yes, great. I think that it can be the bottleneck, I guess, for some. Even I don't know, in plants we have to even have some really other manual steps to prepare the tissue. So could be the bottleneck of the study, because now with the sequencing instrument, which is basically a USB stick, that's easier. But I have a second question. In your air pollution study that you presented, did you find, or probably you didn't check, but did you find pollen DNA? So tree DNA,
which relates to the same question, I guess. So
maybe.
So for the plant part, Christian, very very quickly following up there, right? Yeah, obviously, right, for getting certain DNA out of certain cells, you would need to treat like cells more harshly. I don't have a lot of experience with plants myself. For the aspergillosis project, though, that I showed you, we had to benchmark quite a lot to get actual DNA also from Aspergillus spores, right? So we did a lot of experiments at the beginning in the laboratory, and actually what we ended up doing there was to use like a handheld pistol machine with like small, very tiny glass beads. And we would just like manually do that for 5 minutes for every sample. So I hated it. But with that, we actually reliably got Aspergillus reads out of it. So maybe something like this could also work for plants, whereas I can also, depends on the species probably. And I guess you might also need like an actual heat treatment and so on. But so very good point with DNA extraction, right? DNA extraction is going to influence what we see in the final data set. Now, for our study here, for this first benchmarking for the air microbiome, I was actually not so much interested in all the pollen and also potential fungi that are floating around, because a lot is actually known about fungal species in the air, and I know that obviously at VSL there is a lot of expertise for that, so we were like really more interested on like the bacterial level beyond that, because the question is always, right, like what do you want to use your sequencing efforts for? And I would have feared, especially if it started breaking like pollen cell walls and so on, we might have maybe, just found plants, because the obviously the air microbiome itself is very sparse in the air. Now, this was like really just our first attempt at this. And I do definitely admit, right, that I think one should, to holistically assess the air microbiome, probably use several DNA extraction approaches and then pool the DNA, right? Or prepare different libraries of this DNA and then pool on the library level and then sequence those together. Because the problem is, if we treated those samples really harshly, we would completely shredder all of the DNA of our gram-negative bacteria and so on, and we would lose those. Right. So I think you basically, you would need like several approaches to really reflect the true diversity. But I do think also that's a problem that we have in everything metagenomics wise. Right. But I would be really interested to like treat these samples a bit more harshly and see what else we would find there. Yeah.
Actually, I think as far as I know, a big part of the pollen flying around are already destroyed, as far as I know. So maybe it would not even need to do that. So my question was also, did you actually, with these sequences that you show here, did you actually check in the world of trees, or did you reduce your search basically only to this bacterial thing?
So another question about which I could talk forever, the computation.
Then I stop.
No, I will restrict it to the most important information. So like you will also obviously only find in this sort of data what you provide in your database, which is why, just quickly coming back to that, right, the de novo assembly is such a cool approach, because with de novo assembly, we can potentially get like high confidence evidence that something else is there that's not yet in our databases. But like, I mean, this approach is here, right, they are completely database based, obviously, because we can directly assign them to the taxa. Now here, like we have used different approaches, but we have definitely also included like for example, the entire NCBI-NT database that would also include plants, for example. Now with this, we have like a huge problems, because we might have a lot of false positive mapping, right? If you like provide such a big, like unfiltered database, whereas if we, for example, use a more manually curated database, such as the prokaryotic database of NCBI and so on, you will get a lot of high confidence hits, but probably you are not going to manage to like actually map a lot of your reads to this database. And there we are actually like also working on computational approaches to say that we can use big dirty databases, but we can subsequently filter our mappings depending on the coverage like of the different taxa that we get. Right, like for example, if you imagine you had like one tree species, let's say, and we actually had reads mapping there, but they were all mapping to the very same location of the reference genome. Right, then we can assume that it's actually like a false positive. Either our reads are mapping to the wrong location, or maybe actually the assembly of this tree species itself was not the greatest and it incorporated, some tree microbiome in the assembly. But by looking into these sorts of patterns, you can find out what is likely, like a false positive mapping or not. Yeah, so that's how we are trying to sort of go around this, that we can extend our database, to like any sort of species, including plant species. But yeah, so we would have totally found plants here if they were there, and we didn't find anything.
Thank you.
Perfect, thank you. Well, I guess I've got one as well, then, that I just around the extent to which you're actually sort of moving beyond a kind of, I guess, real-time meta barcoding approaches to something that's genuine real-time metagenomics. You said that what you tend to recover from the nanopore sequencing is very fragmented DNA. Is it just a question of the volume of sequencing and the bioinformatics that you can provide, or do you feel that you're already at a metagenomic analysis stage?
Yeah, so these results that I've shown you here, they are all metagenomics, right? The DNA per se that we get, but that only applies to the air samples, is very fragmented, right? Because of the having a very sparse, like microbiome, and how we are sampling the air with liquid impingement, it's fragmenting it in addition, and so on. So like for this sample type, we just have really fragmented DNA, but it's still from potentially the entire genome of the microorganisms, right? So it's already like a metagenomic approach. And I think it has been like very promising to see, as I said, that we, even with this sort of fragmented reads in the air samples, can build these de novo assemblies. And I don't have the figure here today, but if I showed you, the assemblies for something like that, where, the clinical approach, where we actually can, like when we sequence, we mainly sequence the one pathogen that has been responsible for the disease, then we get like really, really beautiful de novo assemblies out of it, right? So I definitely think it's like promising to see that we can do metagenomics on any level. The robustness obviously differs a bit based on the DNA fragmentation. Does that answer your question?
I think so, yeah. And well, I guess the at heart, what I'm interested in is whether or not you think we can extend this up to the organisms we're interested in. So which, we're talking about larger, more complex genomes typically. Mm-hmm. Is there a practical bottleneck that means that nanopore sequencing might never be the approach that we could use? Or, is it just a question of kind of computational scale that that we could reach the same point?
Mm-hmm. So I think for any sort of like assembly-based approach, right, where you would like to create new genomic data without having to use these incomplete and dirty databases, right? Like I think the most important thing will be long reads. And they are like, not even sequencing accuracy is so important, right? But like having a few long reads that you can then really use to understand the general structure of what of what your genomic data look like, because that will make any sort of downstream assembly more important. So I think like maybe the one shortcoming right now is that we can't get like super long reads yet, right? We couldn't but we can get reads that are like 3-4 megabases long, right? Like one piece. For certain applications, you might even need maybe longer reads, right? If you really wanted to do a telomere to telomere representation, and many of them, right? So I think there's definitely still like some room to further improve the length, because it's just so very helpful for us understanding that our one read comes from one physical DNA piece in our sample, right? I think that's like very, very like very, very valuable information. But I think beyond that, in addition with that, sequencing accuracy in nanopore has like increased a lot, because so what they did in the latest chemistry is they just increased, I have too many pictures, they increased the pore length, of these nanopores. So more bases are in there at the very same time now, which means that every base is seen by more signals in our squiggle signals, so the sequencing accuracy also increased, and it's like over 99% now. I think with that, like the combination of those two things, like should really allow you to do nearly anything you want.
Yeah. Superb. Great, thank you. Would anybody else like to ask something? I think everyone is, holding back. Okay. I see there are one or two beginning to leave as well now. Can I just ask, give you one last chance if there's anything else anybody wanted to follow up with?
Yeah, and obviously also feel free to contact me, right? I can, no, I stopped sharing. I can quickly share contact details again at the end, but like with any questions or if you have applications yourself.
Great. Felix, did you want to follow up with one further?
Well, just perspective-wise, do you think it's, or within which time period would you expect that it is feasible to establish like a large-scale, spatially, temporally, a monitoring system for pathogens, like at least in densely populated areas, maybe where the pressure for pathogen and pandemic responses would be quite large?
Yeah, so I think this will depend a bit on the level of automation that we can achieve, right? Because like as we discussed, I guess already, like a few of the steps already still require some manual processing, like the DNA extraction and so on, which we want to overcome. Yeah, so we would really like to work more and more towards automating so that like any user can just use these devices, like really thinking, like in a few years, having a box that does, for example, air sampling, DNA extraction, library preparation, sequencing, data analysis, all on its own. If you had something like this, I think one could really employ something like this in a more strategic manner at, for example, like locations of high population density, of high traffic, right, at airports and so on, for example, for these sorts of pathogen monitoring. And that's really where we want to go to, and we are actually working on this with our industrial partners at Genomics in the UK at the moment. But I think until then, there's obviously always at the moment at least still going to be a scientist involved, right, in this sort of research. But I do think to like actually create some first One Health data that is maybe focusing on specific pathogens, right? Like also in Switzerland, like certain pathogens that might become problematic or more and more problematic, like brucellosis, like West Nile virus and these sorts of things, right? Like to really think about how can we from different sample types, or which sample types should we even look at, right? But how can we also infer from different sample types how transmission has happened and how mutation has happened? Will just be like, I think, theoretically very interesting to then basically prepare when the automation approaches are there, like to know how to monitor different pathogens. I'm sure there would be a huge difference, for example, between RNA viruses, right, that can spread and mutate so quickly, and potential bacterial pathogens and so on. So I definitely don't have an answer for that, but that's where we would like to go to. But I think we need some like very holistic One Health studies for that first, to see a little bit how powerful our data is as well, to find these connections between different systems.
Maybe a kind of a pooling approach would also be an idea, as it was done here in Switzerland in schools. But you had many samples put into one pool, checked, and then if a pool is positive, then you go into the single samples.
Yeah, or like absolutely, or like what has obviously been done for wastewater surveillance, right? Which is a little bit using the natural pooling effect, I guess.
Yeah, exactly. Yeah.
Something that can be very interesting, or like also with respect to air filters, right? Like there are a lot of air filtering approaches going on anyways, right? Where air filters are being replaced regularly, like in hospitals and so on, right? Where one could also just establish certain sites of monitoring. That's a really good point. Yeah.
Cool. Thanks.
Great. So your path is clear, Felix. You know what to do now.
Mine?
Yeah.
What, get sick?
Okay. I think if there aren't any further questions, then I think we can probably bring things to a close. So it just remains for me to say thanks again, Lara, for a fascinating talk, really interesting, and I'm sure you'll have some follow-ups after this. Can I just remind everybody that there will be a talk next week? So the third seminar is coming up. It will be from Janine Cavender-Barres and Marina Hernandez-Leal. We'll be talking about phenotypes in phylogeography and phylogenetics. So please do come along to that. And otherwise, yeah, we hope to see you there. Thanks again, Lara, and good night, everyone.
Thanks to all of you. Yeah, good night.